Hello everyone, welcome to our discussion on sex determination. In the previous session, we have discussed the first two types of sex determination, which is the chromosomal sex determination and the genic balance theory. Now we are moving on to the third type of sex determination, which is the hormonal influence of sex determination. So, in this, we will see how the hormones are influencing the determination of sex in organisms. So, first is the condition called as free Martin in cattle. So, in the case of cattle, when twin calves of opposite sexes are born, one will be a normal male and the other will be a sterile female with male like traits. This always happens. This always happens when the twin calves of opposite sexes are born. So, this condition is prevalent in cattle, in cows, in all the types of cattle when calves are born which is having opposite sexes. One will be a normal male and the other will be a sterile female with male like traits. And this sterile female is called as free martin. So it is a female and that will be a sterile. And why it becomes sterile is because of the hormonal influence. So through the fused blood vessels of the placenta, what happens is the blood from one embryo with its hormones may enter the bloodstream of the other. And when such event happen, that is when the hormones from the male enters the body of the female, the male hormones influence the development of the female embryo to a male pattern of development. So this is seen in cattle, and such a cattle where the sex of that cattle is determined already. The sex is determined by the chromosome, but the chromosomes also influence the sex. That means it is becoming sterile and it is showing some male like traits because of this particular what you call it as the hormones or the hormonal influence. And because of the influence of the hormone, female embryo or the female particular cattle is shifting its pattern, even though its sex was determined as female genetically, and it is having the genetically determined sex organs, etc. It will show some male traits and it becomes sterile due to hormone influence. And this is what is called as the free margin in cattle, and this is an example of hormonal influence of sex determination. And next is sex reversal in birds. So, in some experiments which was conducted, when early removal of ovary from a genetic female may result in the development of the male comb and plumage and even testes appear. And it was also reported that a hen that was laying eggs ceased to lay egg and developed a male comb plumage and functioned as male and became father to two chickens. And in this particular case, it was reported that this particular hen, its ovary was destroyed by a disease. And when the ovary was destroyed, it was replaced by a testis. It occurred naturally. It was not an experimental work. When the ovary was destroyed by some disease, it was replaced by a testis and it became a male. So this sex reversal is usually seen in birds, and such cases are also reported in pigeons, in amphibians, fishes, and certain lower forms. So this is also an influence because when ovary is destroyed, so a genetically determined sex is there, but when it is being replaced or when the particular hormones are not there, of when the ovary was removed, the hormones which was influencing the female pattern was absent. As a result, male comb started to develop and also the testes started to appear. And that is because of the influence of the male hormones in this case. So that is with the sex reversal in birds. And coming to sex reversal in man. So in man, it is possible that the ovary could be removed from a baby girl and then treated with the male hormones. It would develop masculine appearance. Only masculine appearance can be achieved because, as in the case with higher animals like mammals, when there is a reversal of sex like this, what happens is only secondary sexual characters could be changed. Once formed, the reproductive organs or the reproductive or the external reproductive structures cannot be or cannot be reversed. there could not be complete changeover of the sex organs in man because of the advanced state of development at birth. At birth itself, a complete all the complete development is over before a baby of a human is born. As a result, a complete reversal of the sex is not possible, but the secondary sexual characters can be changed. So the removal of gonads from the adolescents prevent the expression of the secondary sexual characters. So that is also seen in the case with man. So a boy who is castrated, castration means the removal of testes. Castrated before adolescence, fail to develop a beard, the voice. So these are all the secondary sexual characters and other characters of man. And instead, because of the absence, when testis is removed, the testosterone, the hormone is absent. As a result, some female-like characters like voice, hair pattern, etc. appear. So the voice, the hardening of the voice or the bassing or the bass volume of the bass tone of voice in man is due to the or it is due to the changes that happens in adolescence because of the punch of testosterone. But when that testosterone is absent because it is castrated and this castration happens before adolescence, so the voice remain as the high pitched voice of females, as well as the hair pattern also appear in the absence of the testosterone. So the same thing, when the ovary is removed, it retards the mammary gland development. So transplantation of the gonads from one sex to another also produces marked changes in the secondary sexual characters. So only secondary sexual characters can be achieved by such treatments of hormones, etc. In the case of men. The same thing is done in transgenders also. When transgenders they undergo this sex reversal surgeries. So surgeries are performed where if a particular transgender is having if he is genetically a male having a male sex organs, and if he wishes to transform to a and if he's having an inclination towards femaleness because of the hormonal or the hormonal imbalances, etc. If he is having an inclination towards femaleness and he wishes to have a surgery will be performed where castration will be done. That means testes will be removed, and by the surgical process, the sex organs or there is an entire. It is a very complex procedure where the entire sex organ or the external genitalia will be remodeled to the sex which is required, whether from male to female or female to male. The same thing as sex, the surgery is done. And after the surgery, if that particular male he wishes to be female and he has done the surgery, what happens is that particular individual will be under hormonal therapy. Without the hormones, they cannot, because they do not have an ovary in them to produce estrogen, which gives the femaleness. The other thing also, in if a particular female is going to be a man, also the same thing happens because you cannot implant a testis, etc. So a surgery will be performed to remodel their external genitalia, and they will be under hormonal treatment. And hormonal treatment throughout the entire life, they will be under the hormonal treatment. And this hormonal treatment is to attain the secondary sexual characters. For a male, treatment with the female hormones will produce, because overdose of female hormones will produce some feminish or the female like uh, secondary sexual characters, development of breast, then deposition of fat, etc. So that can be achieved. And also the other case with male, providing with testosterone, the masculine appearance, the voice change, etc. Can also be achieved. So this is what is called as sex reversal or the hormonal influence. So in man, the content is that in man you cannot completely change the external genitalia or the sex organs by hormonal influence. Only thing that can be done under hormonal influence of sex determination in man is the secondary sexual characters can be reversed. Coming to the last of sex determination, the fourth type, which is the environmental sex determination. So this is one example of environmental sex determination. There is also other examples like in reptiles, in crocodiles, in turtles, etc., which lay their egg on land. So they dig the particular soil, or the sea floor, or the seashore, if it is a crocodile, whether it is in the sand, they dig and they lay the eggs in the land and they cover it. And incubation is done by inside the soil with the heat of the sun, etc. So what happens is, at a particular critical time, that means the critical time means the time when the sex is determined. The sex will be determined depending upon the temperature, which is at that particular critical time. That means if at that particular critical time of incubation the temperature is high, the eggs will develop into a particular a particular sex. If temperature is high, it will be one type of sex. If temperature is low, it will be all the eggs will give rise to one type of sex. The other, if the temperature is low, it will give rise to another type of sex. But if one is male, the other will be female, and vice versa. So depending upon the species, it might change. So that is also another example of sex determination that is seen in reptiles, where the temperature of incubation at a critical period determines the sex of the offsprings. So this is another example of a marine worm, which is called as Bornelia viridis, which is an excellent example of environmental determined sex. So, when you see this particular organism, the marine worm, there is a great amount of sexual dimorphism between the male and the female. So, what you see here is an attached form. So, this is a female. So, the female is attached to the substratum. It does not move. It is attached to the substratum. It is having a long tube, which is called as a proboscis. And the proboscis, like a bud of the flower, you have the body of the female. So the female ones are larger. It is having a tube to which it is attached to either the stones or to the sea floor, etc. And it is having a very big body. And you can see that the male is a miniature form. It is very smaller, and that reside inside the body of the female. It is very sperm. It is acting as, or it is like a parasite inside the body of the female. So this is the two male and female of Cornelia viridis, and now we'll see how this is determined. How a male is formed as a parasite inside the body of the female. So after fertilization, the eggs develop into a free swimming larva, and this free swimming larva is having the potential to develop into a male or a female. It is having the capacity to develop into a female one with a proboscis, etc., and a body, or it can form a miniature, or it is having the capacity to form a miniature or a parasitic male. And After the free swimming stage, the larva start to settle. So they start to settle at the bottom. And when or those larvae which settles on a free ocean floor, that means if they are setting on a bottom, or on a rock surface, or any substrate which is on the sea floor, they develop a proboscis and they become the female. And if a particular free swimming larva, when it settles, it lands over the proboscis, or the lands over the proboscis of the female. What happens is, such a larva, through the proboscis, it enters the proboscis, it moves through the proboscis and reaches the body of the female, and that becomes a male. That larva will be transformed into a male. So in this, the proboscis of the female produces hormones that will shift the pattern of development into a male type. So here, although the genetic determinants of both sexes are present in the larva, as we said, they have the potential to develop into both the male as well as female. So genetically, they have the capacity to develop into both type, but the environment will direct the development of a particular sex. Environment in the sense, if it is landing directly onto a sea floor or on a substrate, it will form a female by developing a proboscis. If the larva is landing on the body of the female or the proboscis of the female, that proboscis will guide the development of that particular larva into a male type. So with this, we come to the end of this particular topic, which is called as sex determination. In this, we have discussed the four different type of sex determination in animals. One was the chromosomal one, where we discussed the XX, XY type, and which is seen in Drosophila and man, and also the other type, which is the ZZZW type, which is seen in birds, grasshoppers, etc. And we also discussed the type. The second type, which is the gene balance theory, the third one, the hormonal influence of sex determination, and the last one, which is just the environmental sex determination. So, with this, we come to the end of the session. Thanks for hearing.